everyone, in this video we will discuss the question number 35 of GATE 2013. So we are given with two groups, group 1 and group 2. So we need to match the group 1 and group 2. So in the group 1 we are given with RNase P, RNase H, snRNA and CSTF, and in group 2 their function is listed here. So we need to match. So let's go one by one of group 1. So first it comes RNase P. So RNase P is a ribosome. Ribosome are the catalytically active RNA or RNA protein complex where the catalytic activity is solely provided by RNA. So RNase P is actually used in the case of bacteria when they need to process the tRNA. So how does it happen? This is your pre-ribosomal RNA transcript of a bacteria, prokaryotic. So there is 16S rRNA, 23S rRNA, 5S rRNA, and in between 16 and 23S ribosomal RNA gene, there is tRNA gene, which is around of size of 4S. So after that, some modification will be occurred like methylation, pseudo uridinylation. Okay. So after this kind of modifications, these modifications will trigger the cleavage, right? So here after this modification, the cleavage will be starting to happen. So this number 2, if you will see, this number 2 which is at the edge of a tRNA, so that cleavage is mediated by the RNase P. So after the RNase P cleavage, we will get this tRNA which is of course immature tRNA transcript, which is a intermediate. So it will be converted into the mature tRNA after trimming of this portion, right? So this cleavage of tRNA in the pre transcript, so at that number 2 position, that cleavage of the tRNA is mediated by the RNase P, right? So that's why RNase P is playing role in tRNA maturation, and which is a ribosome. Next, in the option Q, it was RNase H. So RNase H is an endonuclease which catalytically cleavage of RNA in RNA DNA hybrid, right? If there is DNA is conjugated or DNA is making hydrogen bond with RNA, then that particular RNA can be cleaved by RNase H. Option R was given as snRNA, which are small nuclear RNA. So what is the role of small nuclear RNA? They have role in the splicing, right? So you can see here there is a intron here, this yellow color thing. Okay, so remove this intron by the splicing process. There is recruitment of some snRNPs, which are small nuclear ribonucleoprotein. So they are the component of the splicing machinery. So they will be recruited. Initially U1 will come, then U2 will come, then U4, U6 will come, then they will form this lariat kind of structure. After that U1 and U4 will be removed, so only U2, U5 and U6 will be there, and this loop will be formed. After that this this loop will be released as a lariat. So the intron will be released as a lariat, so and there will be only 2 exons. So the intron will be spliced out by this splicing machinery, which is composed of small nuclear RNAs, right? So that is the role of snRNA or small nuclear RNA. And the last option was cleavage stimulation factor CSTF, and is required to induce a cleavage at the 3' signaling region from a nascent mRNA, which is followed by addition of a poly A tail at the 3' end of the mRNA. So as we know that in the mRNA modification, one is the addition of the poly A tail at the mRNA 3' end, right? So this cleavage stimulation factor CSTF is required to, first it will introduce a cleavage. So that protein is required to introduce a cleavage at the 3' signaling region of a nascent mRNA. After that cleavage, after that cleavage there will be addition of the poly A tail. So that is the role of cleavage stimulation factor, or CSTF. So now let's see on the groups. So here you can see, uh, if you will go for the option P, which is RNase P. RNase P is associated with the splicing. Sorry, RNase P is So RNase P is ribozyme. As we discussed, RNase P is a ribozyme which plays role in the tRNA cleavage and which is required for the tRNA maturation. So RNase P is a ribozyme. So P will be 3. Okay. Next is option Q, RNase H, which cleaves RNA in a RNA DNA hybrid. So it will go to option 4. So P3Q4. snRNA takes part in splicing, we discussed now. So R2, and CSTF is responsible for polyadenylation. So S1. So the answer will be P3. Yeah. So P3 Q4 R1. Sorry, P3 Q4 R2 S1. So option A will be right. P3 Q4 R2 S1.